Electron transfer to plastocyanin and photosystem I. Plastocyanin PC receives electrons from the cytochrome B6F complex and transfers them to photosystem I PSI. In PSI, the electrons are re-excited by light absorbed by P700 and are transferred to a primary electron acceptor. Reduction of NADP+ to NADPH. The electrons from PSI are passed to ferredoxin FT, which transfers them to NADP+ reductase. Electron transfer to plastocyanin and photosystem I. Plastocyanin PC receives electrons from the cytochrome B6F complex and transfers them to photosystem I PSI. In PSI, the electrons are re-excited by light absorbed by P700 and are transferred to a primary electron acceptor. Reduction of NADP+ to NADPH. The electrons from PSI are passed to ferredoxin FT, which transfers them to NADP+ reductase. NADP+ reductase catalyzes the reduction of NADP+ to NADPH using the electrons provided by ferredoxin. Chemiosmotic ATP synthesis. The proton gradient established across the thylakoid membrane, high concentration of protons in the lumen creates a proton motive force PMF. ATP synthase utilizes this gradient to synthesize ATP from ADP and pi by chemiosmosis. Importance of electron transport in photosynthesis energy conversion. The electron transport chain converts light energy into chemical energy in the form of ATP and NADPH. These energy carriers are critical for driving the Calvin cycle, where carbon fixation occurs. Oxygen evolution. The splitting of water in photosystem 2 leads to the release of oxygen as a byproduct, which is vital for sustaining life on Earth. Proton gradient and ATP synthesis. The proton gradient generated during electron transport is essential for the production of ATP, which provides the energy required for the Calvin cycle and other cellular processes. Previous year CSIR net questions. CSIR net June 2020. Question. The enzyme NADP+ reductase in photosynthesis is responsible for a splitting water molecules. B reducing NADP+ to NADPH. C catalyzing the reduction of oxygen to water. D transfer of electrons from plastocyanin to PSI. Answer. B reducing NADP+ to NADPH. NADP+ reductase reduces NADP+ to NADPH by utilizing electrons from PSI. CSIR net December 2018. Question. Which of the following is responsible for transferring electrons between photosystem 2 and photosystem I? A. Plastocyanin. B. Cytochrome B6F. C. Plastoquinone. D. Ferredoxin. Answer. A. Plastocyanin. Plastocyanin transfers electrons from cytochrome B6F to photosystem I. CSIR net June 2017. Question. The proton motive force PMF generated during electron transport in photosynthesis is used to a split water molecules. B pump electrons through the cytochrome B6F complex. C synthesize ATP via ATP synthase. D reduce NADP+ to NADPH. Answer. C synthesize ATP via ATP synthase. The proton gradient established by electron transport is used by ATP synthase to produce ATP. This lecture provides a detailed and exam-focused understanding of electron transport in photosynthesis, with a strong emphasis on key concepts, mechanisms, and previous year questions to help in CSIR net JRF life sciences preparation. It covers the fundamental mechanisms, proteins involved, and significance of electron transport in photosynthesis, ensuring thorough preparation for the exam. You said. Photoprotective mechanisms. ChatGPT said. Here's a highly detailed lecture on photoprotective mechanisms tailored for CSIR net JRF life sciences preparation. This lecture is designed to focus on key concepts, important points, and exam-oriented details from base to advanced level as per your requirements. Photoprotective mechanisms in photosynthesis. Photoprotection refers to the set of mechanisms in plants that help mitigate the damage caused by excess light during photosynthesis. When plants are exposed to light intensities that exceed the photosynthetic capacity, reactive oxygen species, ROS, are produced, leading to potential damage to photosynthetic apparatus. Photoprotective mechanisms help in preventing or repairing such damage, ensuring the integrity of photosystem complexes and the continuation of the photosynthetic process. Mechanisms of photoprotection. Photoprotective mechanisms are broadly classified into non-photochemical quenching (NPQ) repair mechanisms and antioxidant systems. These mechanisms work together to safeguard the plant's photosynthetic machinery. 1. Non-photochemical quenching (NPQ). Non-photochemical quenching is the primary mechanism for dissipating excess absorbed light energy as heat, thereby preventing the formation of ROS. Mechanism. NPQ involves the xanthophyll cycle, which is responsible for dissipating excess light energy in the form of heat. Under high light conditions, the xanthophylls such as zeaxanthin and bilaxanthin are interconverted, which leads to a reduction in the excitation energy that would otherwise be used to generate reactive oxygen species ROS. Bilaxanthin to zeaxanthin conversion. In the presence of excess light, bilaxanthin is converted to zeaxanthin through the action of bilaxanthin to oxidase (BDD). Zeaxanthin helps to quench excess energy in the photosystem by dissipating it as heat. Role of heat dissipation. The heat dissipation reduces the potential for ROS formation by preventing excessive excitation of the photosystem reaction centers. The process is regulated by protein components such as PSII and LHCII, light harvesting complex 2. Significance in photoprotection. Non-photochemical quenching plays a crucial role in protecting plants from light-induced damage by regulating the absorption and utilization of light energy. 2. Antioxidant defense mechanisms. Plants use a complex network of antioxidants to detoxify the reactive oxygen species, ROS, that can cause oxidative damage to cellular components. Superoxide dismutase, SOD. Superoxide dismutase catalyzes the conversion of superoxide radicals O2 into hydrogen peroxide, H2O2, which is then further detoxified. Ascorbic peroxidase, APX, and glutathione peroxidase, GPX. These enzymes play a key role in the conversion of hydrogen peroxide into water, thereby protecting the cells from oxidative damage. Catalase, CAT. Catalase is an enzyme that helps convert hydrogen peroxide into water and oxygen, providing an additional line of defense against ROS-induced damage. Glutathione, GSH. Glutathione is a critical antioxidant that plays a role in neutralizing reactive oxygen species and maintaining cellular redox balance. Role of antioxidants in photoprotection. Antioxidant enzymes are located in the chloroplasts, mitochondria, and cytosol, providing protection against the oxidative stress caused by excess light absorption. 3. Repair mechanisms of photosystem 2 PSII. Excess light can damage the photosystem 2 PSII complex. Plants possess several repair mechanisms to protect PSII and restore its functionality. D1 protein repair. Photosystem 2 contains a protein called D1, which is particularly sensitive to photodamage under high light conditions. The D1 protein is continuously synthesized and degraded to prevent permanent damage to PSII. When the D1 protein is damaged due to excess light, it is degraded, and a new D1 protein is synthesized in the thylakoid membrane. PSII turnover. PSII turnover is essential for maintaining efficient photosynthetic activity, especially under fluctuating light conditions. The degradation of damaged PSII components and the synthesis of new ones are critical for the recovery of photosynthetic efficiency. Regulation of the repair cycle. The repair of PSII is facilitated by PSII repair cycle proteins, such as FTSH protease, which degrade the damaged D1 protein, and protein synthesis machinery that synthesizes new proteins to replace the damaged ones. Significance of PSII repair in photoprotection. This repair mechanism ensures that plants can adapt to varying light conditions and maintain optimal photosynthetic performance even under stress. 4. Stomatal regulation. Another photoprotective mechanism involves stomatal regulation, which helps reduce the absorption of excess light energy by controlling the CO2 intake. Stomatal closure. Under conditions of high light and water stress, stomata close to reduce the loss of water and to prevent the absorption of excess light energy. This regulation helps in minimizing photodamage. Role of abscisic acid, ABA. ABA is a key signaling molecule that induces stomatal closure during water deficit conditions. This indirect photoprotective mechanism ensures that the plant does not overabsorb light, which could lead to the production of damaging ROS. Significance of photoprotective mechanisms. Photoprotective mechanisms are crucial for maintaining photosynthetic efficiency. They help maintain optimal photosynthetic performance, particularly under fluctuating environmental conditions like light intensity and temperature. Preventing oxidative damage. Excess light can produce ROS, which can damage the photosynthetic apparatus PSI, lipids, proteins, and DNA. Photoprotective mechanisms ensure the neutralization of ROS, thus preventing cellular damage. Survival under stress. These mechanisms enhance the ability of plants to tolerate stress conditions such as high light intensity, drought, and temperature fluctuations, which could otherwise impair growth and productivity. Previous year CSIR net questions. CSIR net June 2020. Question: which of the following xanthophylls is involved in the non-photochemical quenching of excess light energy? A. Zeaxanthin. B. Leucine C. Beta-carotene D. Bilaxanthin. Answer: A. Zeaxanthin. Zeaxanthin is involved in non-photochemical quenching by dissipating excess light energy as heat. CSIR net December 2019. Question: the photoprotective mechanism of NPQ involves the interconversion of which of the following pigments? A chlorophyll to chlorophyll B. B. Bilaxanthin. Question: which of the following xanthophylls is involved in the non-photochemical quenching of excess light energy? A. Zeaxanthin. B. Leucine C. Beta-carotene D. Bilaxanthin. Answer: A. Zeaxanthin. Zeaxanthin is involved in non-photochemical quenching by dissipating excess light energy as heat. CSIR net December 2019. Question: the photoprotective mechanism of NPQ involves the interconversion of which of the following pigments? A chlorophyll to chlorophyll B. B. Bilaxanthin to zeaxanthin. C. Leucine to zeaxanthin. D. Phytochrome to cryptochrome. Answer: B. Bilaxanthin to zeaxanthin. The interconversion of bilaxanthin to zeaxanthin is a key step in non-photochemical quenching, which protects plants from excess light. CSIR net June 2018. Question: which of the following is responsible for the repair of the photosystem to under high light conditions? An ADP plus reductase. BFTSH protease C ATP synthase D cytochrome B6F complex. Answer: BFTSH protease. FTSH protease is involved in the degradation of the damaged D1 protein in PSI during the repair process. Conclusion: photoprotective mechanisms in plants are essential for maintaining the balance between light absorption and energy dissipation. These mechanisms, ranging from non-photochemical quenching (NPQ) antioxidant systems to PSI repair mechanisms, ensure that plants can survive under variable environmental conditions and prevent damage to the photosynthetic machinery. Understanding these mechanisms is crucial for CSIR net JRF Life Sciences aspirants to excel in the exam. This lecture provides high-quality, exam-oriented content designed to equip you with the necessary knowledge to crack the CSIR net JRF Life Sciences exam. You said CO2 fixation. ChatGPT said. Here's a highly detailed lecture on CO2 fixation, tailored for CSIR net JRF Life Sciences preparation. This lecture covers key concepts, important points, and exam-oriented details from base to advanced level, ensuring you have everything you need to excel in the exam. CO2 fixation in photosynthesis. CO2 fixation refers to the process by which carbon dioxide (CO2) from the atmosphere is converted into organic molecules during the light-independent reactions of photosynthesis. It is the foundational step in the synthesis of glucose and other organic compounds, which are essential for plant growth and metabolism. The Calvin cycle is the primary biochemical pathway for CO2 fixation in most plants, though there are variations like C4 and CAM pathways in certain plants adapted to specific environmental conditions. 1. The Calvin cycle C3 pathway. The Calvin cycle is the most common pathway for CO2 fixation in C3 plants. It occurs in the stroma of chloroplasts and involves the fixation of CO2 into an organic molecule, followed by a series of reactions that regenerate ribulose bisphosphate, the molecule responsible for CO2 fixation. Mechanism of CO2 fixation in the Calvin cycle: carbon fixation. CO2 is initially fixed by the enzyme ribisco, ribulose 1,5-bisphosphate carboxylase oxygenase. In this step, CO2 reacts with ribulose 1,5-bisphosphate rub, a 5-carbon sugar, producing an unstable 6-carbon compound that immediately breaks down into two molecules of 3-phosphoglycerate, 3-PGA, a 3-carbon compound. Reduction phase. In this phase, 3-PGA is converted into glyceraldehyde 3-phosphate G3P through a two-step process involving ATP and NADPH generated during the light-dependent reactions. This phase uses the energy from light to reduce 3-PGA into G3P. Regeneration of rub. Some of the G3P molecules exit the cycle and are used to synthesize sugars, while the remaining G3P molecules are used to regenerate rub, ensuring the cycle can continue. This regeneration requires additional ATP molecules. End products. The Calvin cycle results in the production of glucose, fructose, and other carbohydrates that the plant can use for energy and growth. 2. C4 pathway. C4 photosynthesis. The C4 pathway is found in plants that are adapted to hot, dry environments, such as corn, sugarcane, and millets. This pathway helps plants overcome the inefficiencies of the C3 pathway, particularly under high light intensity and temperature conditions. Mechanism of CO2 fixation in C4 plants. CO2 fixation in mesophil cells. In C4 plants, CO2 is initially fixed by the enzyme PEP carboxylase, phosphoenolpyruvate carboxylase in the mesophil cells. This enzyme has a higher affinity for CO2 than ribisco and fixes CO2 to phosphoenolpyruvate, PEP, forming oxaloacetate, OAA, a 4-carbon compound. Formation of malate or aspartate. Oxaloacetate is then converted into malate or aspartate, which are transported into the bundle sheath cells. CO2 release and fixation by ribisco. In the bundle sheath cells, malate is decarboxylated to release CO2, which is then fixed by ribisco in the Calvin cycle to form 3-PGA. Advantages of C4 pathway. The C4 pathway minimizes photorespiration, a wasteful process in which ribisco fixes oxygen instead of CO2. This adaptation allows C4 plants to perform photosynthesis more efficiently under high temperatures and light intensities. 3. CAM pathway. Crassylation acid metabolism. The CAM pathway is found in plants like cacti, succulents, and pineapple, which are adapted to arid conditions with limited water availability. Mechanism of CO2 fixation in CAM plants. Nighttime CO2 fixation. During the night, CAM plants open their stomata and fix CO2 into oxaloacetate via PEP carboxylase, similar to the C4 pathway. The fixed CO2 is stored as malate in vacuoles. Daytime CO2 release in Calvin cycle. During the day, when the stomata are closed to minimize water loss, malate is decarboxylated to release CO2 in the mesophil cells, where it is then fixed by ribisco in the Calvin cycle. Advantages of CAM pathway. The CAM pathway allows plants to conserve water by fixing CO2 at night when the temperature is lower and water loss via transpiration is minimized. 4. Regulation of CO2 fixation. The process of CO2 fixation is tightly regulated at multiple levels to ensure efficient photosynthesis. Key regulatory mechanisms. Rubisco activation. Rubisco is activated by Mg2+ ions and CO2 binding. It is also regulated by light, as the Calvin cycle depends on the ATP and NADPH produced during the light-dependent reactions. pH and ion regulation. The pH of the stroma increases during light reactions, which activates the Calvin cycle enzymes. Additionally, the concentration of inorganic phosphate and ATP is critical for regulating the cycle. Carbon concentrating mechanisms, CCMs. In C4 and CAM plants, the concentration of CO2 is elevated in specific cells, e.g., bundle sheath cells in C4 plants and mesophil cells in CAM plants, ensuring efficient fixation by rubisco. Significance of CO2 fixation. CO2 fixation is the primary mechanism by which plants convert inorganic carbon into organic compounds, forming the foundation of the food chain. The sugars produced during this process are vital for energy production, growth, and development in plants. The efficiency of CO2 fixation directly impacts photosynthetic productivity and, in turn, agricultural yield and biomass production. Previous year CSIR net questions. CSIR net June 2021. Question: which enzyme is responsible for the fixation of CO2 in C4 plants? A. Rubisco. B. PEP carboxylase. C. Rubisco activates D aldolase. Answer. B. PEP carboxylase. PEP carboxylase fixes CO2 into oxaloacetate in the mesothelial cells of C4 plants. CSIR net December 2019. Question. In which of the following pathways is CO2 fixed at night to form malate? A. C3 pathway. B. C4 pathway. C. CAM pathway. D. Both C4 and C3 pathways. Answer. C. CAM pathway. CAM plants fix CO2 at night, storing it as malate, and release it during the day for fixation by Rubisco in the Calvin cycle. CSIR net June 2018. Question. The Calvin cycle is most active in which part of the plant? A. Root. B. Stem. C. Leaf. D. Flower. Answer. C. Leaf. The Calvin cycle occurs in the stroma of chloroplasts, which are abundant in the leaf. Conclusion. CO2 fixation is a central process in photosynthesis, ensuring the synthesis of organic molecules that support plant growth. From the Calvin cycle C3 pathway to C4 and CAM pathways, plants have evolved diverse mechanisms to optimize CO2 fixation under different environmental conditions. Understanding these mechanisms is crucial for the CSIR net JRF Life Sciences exam, as it is a fundamental topic in plant physiology and biochemistry. You said C3 pathways. ChatGPT said C3 pathway Calvin cycle in photosynthesis. The C3 pathway, also known as the Calvin cycle, is the most common pathway of carbon fixation in plants. It occurs in the stroma of the chloroplast and is crucial for converting carbon dioxide (CO2) into organic molecules that are used for energy, growth, and metabolism. The C3 pathway is so named because the first stable product formed is a 3-carbon compound, 3-phosphoglycerate, 3-PGA. This pathway is utilized by C3 plants, which include most plants, such as wheat, rice, soybeans, and tobacco. Steps of the C3 pathway Calvin cycle. The Calvin cycle consists of 3 main stages: carbon fixation, reduction, and regeneration of Rub. Let's break down each step in detail. 1. Carbon fixation. Rubisco ribulose 1,5-bisphosphate carboxylase oxygenase is a central enzyme in the C3 pathway. It is responsible for fixing CO2 into an organic molecule. CO2 from the atmosphere enters the plant through the stoma and diffuses into the chloroplasts of the mesophyll cells. In the stroma, CO2 combines with a 5-carbon molecule, ribulose 1,5-bisphosphate Rub, in the presence of Rubisco, forming an unstable 6-carbon compound. This unstable 6-carbon compound immediately splits into 2 molecules of 3-phosphoglycerate, 3-PGA, each having 3 carbon atoms. This step is called carbon fixation because carbon from CO2 is now incorporated into an organic molecule. 2. Reduction phase. In this phase, the 2 molecules of 3-PGA formed during carbon fixation are converted into glyceraldehyde 3-phosphate G3P, A3 carbon sugar. The conversion of 3-PGA to G3P requires energy in the form of ATP from the light-dependent reactions, and NADPH also produced during the light-dependent reactions. Specifically, 3-PGA is phosphorylated by ATP, forming 1,3-bisphosphoglycerate, which is then reduced by NADPH to form G3P. This step is crucial for producing sugars. 3. Regeneration of Rub. After some of the G3P molecules exit the cycle to form glucose and other sugars, the remaining G3P molecules are used to regenerate Rub, allowing the cycle to continue. This step requires additional ATP. Through a series of enzyme-mediated steps, G3P molecules are rearranged to regenerate Rub, a 5-carbon molecule that can now accept more CO2. The regeneration of Rub is essential for the continuation of the Calvin cycle and ensures that the cycle can keep running. Net reaction of the Calvin cycle C3 pathway. The overall reaction of the Calvin cycle can be summarized as 6 CO2 plus 18 ATP plus 12 NADPH plus H2O right pointing arrow C 6 H12O6 plus 18 ATP plus 18 PI plus 12 NADP plus 6 CO2 plus 1 8 ATP plus 1 2 NADPH plus H2 O right pointing arrow C6 H12O6 plus 1 8 ATP plus 1 8 pi plus 12 NADP plus. This represents the fixation of 6 molecules of CO2 utilizing ATP and NADPH to form glucose C6H12O6, a simple sugar that serves as an energy source for the plant. Importance of the C3 pathway. The C3 pathway is the foundation of photosynthesis in most plants, making it crucial for the production of organic compounds that sustain life on Earth. The sugars produced through the Calvin cycle serve as building blocks for other essential plant compounds, such as starch, cellulose, and proteins. However, the C3 pathway is not without its limitations. Under conditions of high temperature, light intensity, and water stress, photorespiration becomes a problem. In photorespiration, Rubisco reacts with O2 instead of CO2, leading to the production of harmful byproducts. This inefficiency in CO2 fixation results in a loss of energy and carbon. Photorespiration and its impact on the C3 pathway. Photorespiration occurs when Rubisco fixes oxygen, O2, instead of carbon dioxide, CO2. This process is detrimental to the plant because it consumes energy and reduces the efficiency of photosynthesis. The phenomenon is more pronounced at higher temperatures, where the affinity of Rubisco for O2 increases, especially when the stomata are close to conserved water. The process of photorespiration produces phosphoglycolate, which has to be recycled through a series of reactions involving the peroxisome and mitochondria, ultimately leading to the loss of fixed carbon and nitrogen. This makes C3 plants less efficient under high temperature and drought conditions. C3 pathway versus C4 pathway. The C4 pathway is an adaptation to counteract photorespiration. C4 plants, such as corn and sugarcane, separate carbon fixation in the Calvin cycle into different cells, mesophyll and bundle sheath cells, which allows them to maintain a higher concentration of CO2 around Rubisco, thus minimizing photorespiration. C4 plants are more efficient in hot and arid climates compared to C3 plants. Previous year CSIR net questions. CSIR net June 2020. Question. In the Calvin cycle, the first stable product formed after the fixation of CO2 is A3-phosphoglycerate. B-glyceraldehyde 3-phosphate C-ribulose 1, 5-bisphosphate D-fructose 1, 6-bisphosphate answer, A3-phosphoglycerate. The first stable product formed in the Calvin cycle after CO2 fixation is 3-phosphoglycerate, a 3-carbon molecule. CSIR net December 2018. Question. The enzyme that catalyzes the fixation of CO2 in the Calvin cycle is ATP synthase. B. Rubisco C-PEP carboxylase D-pyruvate kinase answer, B. Rubisco. Rubisco is the enzyme responsible for the fixation of CO2 in the Calvin cycle. CSIR net June 2019. Question. The process of photorespiration occurs when Rubisco fixes oxygen instead of CO2. B. CO2 instead of oxygen C. Nitrogen instead of oxygen D. CO2 and nitrogen answer, oxygen instead of CO2. In photorespiration, Rubisco fixes oxygen instead of carbon dioxide, leading to a loss of carbon and energy. Conclusion. The C3 pathway is the primary mode of CO2 fixation in plants, critical for the synthesis of carbohydrates. Despite being widely utilized, this pathway suffers from inefficiencies under high temperatures due to photorespiration. Understanding the Calvin cycle, its steps, and its limitations is essential for CSIR net JRF Life Sciences preparation. By grasping the intricacies of the C3 pathway, you can confidently answer questions related to photosynthesis and plant physiology in the exam. You said C4 pathways. ChatGPT said C4 pathway C4 photosynthesis. The C4 pathway, or C4 photosynthesis, is an adaptive mechanism used by certain plants to minimize photorespiration and enhance the efficiency of carbon fixation in conditions of high temperature, light intensity, and limited water availability. This pathway is found in C4 plants like maize, sugarcane, millets, and sorghum, which are typically grown in hot and dry environments. Unlike C3 plants, which directly fix CO2 into a 3-carbon compound, C4 plants initially fix CO2 into a 4-carbon compound, thus the name C4 pathway. The primary goal of the C4 pathway is to concentrate CO2 around the Rubisco enzyme, minimizing its interaction with O2, and hence reducing the inefficiencies caused by photorespiration. Steps of the C4 pathway. The C4 pathway can be broken down into 2 major phases, carbon fixation and carbon release, with the crucial distinction that C4 plants use a spatial separation of the processes of CO2 fixation in the Calvin cycle. These processes occur in 2 different types of cells, mesophyll cells and bundle sheath cells. 1. Carbon fixation in mesophyll cells. PEP carboxylase phosphoenolpyruvate carboxylase is the enzyme that initiates carbon fixation in the mesophyll cells. CO2 is first absorbed by the mesophyll cells, where it reacts with phosphoenolpyruvate PEP, a 3-carbon compound, in the presence of PEP carboxylase. This reaction forms oxaloacetate, a 4-carbon compound, which is the first stable product of the C4 pathway. Oxaloacetate is then converted into malate or aspartate, which are also 4-carbon compounds, depending on the plant species. These compounds are transported from the mesoscale cells to the bundle sheath cells. 2. Decarboxylation in bundle sheath cells. Once the malate or aspartate reaches the bundle sheath cells, which surround the vascular tissue, it is decarboxylated by enzymes such as malic enzyme. This decarboxylation process releases CO2, which is now at a higher concentration in the bundle sheath cells. This concentrated CO2 is then available for fixation by Rubisco in the Calvin cycle. The Calvin cycle operates similarly to that in C3 plants, but because the CO2 concentration is higher in the bundle sheath cells, Rubisco is less likely to bind with O2, thus reducing photorespiration. The C3 pathway Calvin cycle in the bundle sheath cells then produces glyceraldehyde 3-phosphate G3P, which is used for the synthesis of sugars and other organic molecules. 3. Regeneration of PEP. After the decarboxylation process, the 3-carbon products like pyruvate produced in the bundle sheath cells are transported back to the mesoscale cells. In the mesoscale cells, pyruvate is converted into PEP by the enzyme pyruvate phosphate digonase PPDK, which requires ATP. This regeneration of PEP allows the cycle to continue, facilitating the ongoing fixation of CO2 into the 4-carbon compounds in the mesoscale cells. Key features of the C4 pathway. Spatial separation of reactions. The C4 pathway separates the processes of CO2 fixation in the Calvin cycle into different cells. Carbon fixation occurs in the mesoscale cells, while the Calvin cycle C3 cycle occurs in the bundle sheath cells. This separation helps concentrate CO2 around Rubisco and minimizes photorespiration. PEP carboxylase. The enzyme PEP carboxylase is highly efficient at fixing CO2, even at low concentrations. It has a very low affinity for oxygen, which is advantageous under high light intensity and temperature, as it reduces the chance of photorespiration. Higher water use efficiency. The C4 pathway allows plants to efficiently use water by maintaining closed stomata during the day to conserve water while still achieving high rates of CO2 fixation. This is especially beneficial in hot, dry climates. Advantages of the C4 pathway. Reduced photorespiration. The spatial separation of CO2 fixation in the Calvin cycle in C4 plants helps reduce the inefficiency caused by photorespiration, which is more prominent in C3 plants under hot and dry conditions. Enhanced carbon fixation. The concentration of CO2 in the bundle sheath cells around Rubisco increases the efficiency of photosynthesis in conditions of high light intensity, temperature, and drought. Higher water use efficiency. The C4 pathway allows plants to conserve water by minimizing water loss through transpiration, which is particularly important in arid environments. C4 pathway versus C3 pathway. CO2 fixation. The C3 pathway fixes CO2 directly into a 3-carbon compound, 3-PGA, while the C4 pathway first fixes CO2 into a 4-carbon compound, oxaloacetate. Photorespiration. C3 plants suffer from photorespiration, especially under high temperature and light conditions. In contrast, C4 plants minimize photorespiration by concentrating CO2 around Rubisco in the bundle sheath cells. Energy cost. The C4 pathway requires additional energy, ATP, to regenerate PEP and transport malate aspartate between cells. However, this energy cost is offset by the enhanced efficiency of carbon fixation and reduced loss due to photorespiration. Previous year CSIR net questions. CSIR net June 2020. Question. In C4 photosynthesis, the enzyme responsible for fixing CO2 into a 4-carbon compound is a Rubisco. B-PEP carboxylase C-pyruvate kinase D-phosphoglycerate kinase. Answer. B-PEP carboxylase. PEP carboxylase fixes CO2 into a 4-carbon compound, oxaloacetate, in C4 plants. CSIR net December 2019. Question. The C4 pathway differs from the C3 pathway by A. Using a higher concentration of CO2 in the bundle sheath cells, B. Using oxygen instead of CO2 in the Calvin cycle, C. Fixing CO2 in mesoscale cells, not bundle sheath cells, C. Regenerating while using ATP in the mesoscale cells. Answer. A. Using a higher concentration of CO2 in the bundle sheath cells, the C4 pathway concentrates CO2 in the bundle sheath cells, reducing photorespiration. CSIR net June 2018. Question. Question. Which of the following is a key feature of C4 photosynthesis? Question. The C4 pathway differs from the C3 pathway by A. Using a higher concentration of CO2 in the bundle sheath cells, B. Using oxygen instead of CO2 in the Calvin cycle, C. Fixing CO2 in the mesoscale cells, not bundle sheath cells, C. Regenerating while using ATP in the mesoscale cells. Answer. A. Using a higher concentration of CO2 in the bundle sheath cells, the C4 pathway concentrates CO2 in the bundle sheath cells, reducing photorespiration. CSIR net June 2018. Question. Which of the following is a key feature of C4 photosynthesis? A carbon fixation occurs in the cytoplasm. B. It involves spatial separation of carbon fixation in the Calvin cycle. C. The enzyme Rubisco fixes carbon in the mesoscale cells. D. It produces glucose directly in the mesoscale cells. Answer. B. It involves spatial separation of carbon fixation in the Calvin cycle. In C4 plants, carbon fixation occurs in the mesoscale cells, and the Calvin cycle occurs in the bundle sheath cells. Conclusion. The C4 pathway is a vital adaptation for plants growing in hot, arid conditions, as it efficiently fixes carbon while minimizing the inefficiencies caused by photorespiration. By utilizing PEP carboxylase and spatial separation of the processes of carbon fixation in the Calvin cycle, C4 plants are more efficient in terms of water and carbon use, making them better suited for high light, high temperature environments. Understanding the intricacies of the C4 pathway is crucial for excelling in CSIR net JRF Life Sciences, especially for questions related to photosynthesis and plant physiology. You said, CAM pathways. ChatGPT said. CAM pathway cresylation acid metabolism. Cresylation acid metabolism CAM is a specialized photosynthetic pathway used by certain plants, particularly those found in arid, semi-arid, and dry environments. CAM plants include cacti, succulents, pineapple, and agave. The primary advantage of CAM photosynthesis is its ability to fix CO2 during the night, when transpiration rates are low, and stored as organic acids for use during the day, thereby minimizing water loss. This mechanism allows CAM plants to survive in extremely hot and dry conditions where water conservation is critical. The CAM pathway is an adaptation to arid climates where high temperatures and scarce water resources make traditional photosynthesis inefficient. While similar to C4 photosynthesis in some aspects, CAM photosynthesis is distinguished by its temporal separation of the carbon fixation process in the Calvin cycle. In contrast to the C4 pathway, which spatially separates these processes into different types of cells, mesoscale and bundle sheath cells, CAM plants perform both carbon fixation and the Calvin cycle within the same cell but at different times of the day. Steps of the CAM pathway. The CAM pathway involves two key phases: night phase carbon fixation and day phase Calvin cycle, with the crucial distinction being that CAM plants fix CO2 at night and perform the Calvin cycle during the day. 1. Night phase carbon fixation. During the night, stomata of CAM plants open to take in CO2. At night, temperatures are lower and water loss through transpiration is minimized. The PEP carboxylase enzyme fixes the CO2 into a 4-carbon compound, similar to what occurs in the C4 pathway. The CO2 is fixed into oxaloacetate, which is subsequently converted into malate or aspartate, both of which are 4-carbon organic acids. These organic acids are stored in vacuoles of the plant cells until the day phase, effectively storing CO2 in the form of malic acid, mainly malate or aspartate. 2. Day phase Calvin cycle. During the day, the stomata close to conserve water as temperatures rise. The stored organic acids, malate, aspartate, are released from the vacuoles and decarboxylated to release CO2 in the cytoplasm. This released CO2 is then available for fixation by Rubisco in the Calvin cycle, where it is incorporated into 3-carbon compounds like 3-phosphoglycerate, 3-PGA. The Calvin cycle proceeds as usual, producing glyceraldehyde 3-phosphate G3P, which is used for the synthesis of sugars and other organic molecules. The CAM pathway thus separates the processes of carbon fixation in the Calvin cycle based on time rather than location. CO2 fixation happens at night, and the Calvin cycle takes place during the day. Key features of the CAM pathway. Temporal separation. The primary feature of CAM photosynthesis is the temporal separation of the carbon fixation and the Calvin cycle, with carbon fixation occurring during the night and the Calvin cycle operating during the day. This helps CAM plants conserve water by minimizing transpiration. PEP carboxylase. Like C4 plants, CAM plants utilize the enzyme PEP carboxylase to fix CO2 into 4 carbon compounds oxaloacetate, malate, or aspartate during the night, a process that is highly efficient even in low CO2 conditions. Stomatal opening. Stomata open at night to take in CO2, reducing the loss of water through transpiration. During the day, the stomata closes to conserve water while CO2 is released from stored organic acids. Water use efficiency. CAM plants are highly efficient in their use of water. The ability to fix carbon at night allows them to reduce water loss by limiting transpiration during the hottest parts of the day. Advantages of the CAM pathway. Water conservation. The key benefit of CAM photosynthesis is its ability to minimize water loss in arid environments. By opening stomata at night, CAM plants can fix CO2 without losing significant amounts of water. High water use efficiency. CAM plants can thrive in extremely dry conditions because they can continue to photosynthesize during the day while keeping their stomata closed. This enhances their water use efficiency, which is crucial for survival in water-scarce habitats. Adaptation to hot and dry environments. CAM photosynthesis is particularly useful in environments with high daytime temperatures, low humidity, and scarce water resources, allowing plants to optimize carbon fixation while avoiding dehydration. CAM versus C4 pathway. Time versus space separation. The primary difference between C4 and CAM pathways lies in their separation of carbon fixation and the Calvin cycle. In C4 plants, carbon fixation and the Calvin cycle are spatially separated into different types of cells, mesophyll and bundle sheath cells. In CAM plants, the separation is temporal, with carbon fixation occurring at night and the Calvin cycle occurring during the day, all within the same cell. Energy requirements. The C4 pathway requires more energy because it involves two different types of cells and a more complex mechanism for transporting metabolites. CAM plants, however, only require energy for the conversion of stored malate to CO2 during the day. Previous year CSIR net questions. CSIR net June 2020. Question. In CAM photosynthesis, the organic acid that is typically stored overnight and used for CO2 release during the day is oxaloacetate. Bean malate C aspartate D-phosphoenol pyruvate. Answer. Bean malate. Malate is the primary organic acid that stores CO2 overnight in CAM plants. CSIR net December 2018. Question. CAM plants open their stomata. A during the day to take in CO2, B during the night to take in CO2, C during the night to release O2, D continuously to take in CO2. Answer. B during the night to take in CO2. CAM plants open stomata at night to minimize water loss while absorbing CO2. CSIR net December 2017. Question. CAM plants fix carbon into 4 carbon compounds during the night, which is later decarboxylated during the day to release CO2. The enzyme responsible for the initial CO2 fixation is Rubisco. B-PEP carboxylase C malate enzyme dephosphoglycerate kinase. Answer. B-PEP carboxylase. PEP carboxylase is responsible for fixing CO2 into 4 carbon compounds in CAM plants. Conclusion. The CAM pathway is a remarkable adaptation that allows plants to survive in hot, dry, and arid environments. By opening stomata only at night to fix CO2, these plants minimize water loss and enhance water use efficiency, making them well suited for challenging climatic conditions. The pathway's temporal separation of carbon fixation and the Calvin cycle, along with the use of PEP carboxylase for CO2 fixation, distinguishes it from both the C3 and C4 pathways. Understanding the mechanisms and advantages of CAM photosynthesis is crucial for CSIR net JRF life sciences students, particularly for questions related to plant physiology, photosynthesis, and adaptation to environmental stress. You said. Respiration and photorespiration. ChatGPT said. Respiration and photorespiration. Respiration. Respiration in plants is a fundamental metabolic process that provides the necessary energy for growth, development, and various physiological activities. It involves the breakdown of organic molecules like glucose to produce ATP, adenosine triphosphate, which cells use as a primary energy source. This process occurs in the mitochondria and can be classified into aerobic respiration and anaerobic respiration depending on the availability of oxygen. 1. Aerobic respiration. Aerobic respiration is the most common form of respiration in plants, occurring in the presence of oxygen. It involves the complete breakdown of glucose into carbon dioxide and water, releasing energy in the form of ATP. The process of aerobic respiration can be divided into 3 stages. Glycolysis location, cytoplasm. Process. Glucose 6 carbon molecule is converted into 2 molecules of pyruvate, 3 carbon molecules producing a small amount of ATP and NADH. Krebs cycle, citric acid cycle, location, mitochondrial matrix. Process. Pyruvate is further oxidized to release CO2, and high energy electrons are transferred to carrier molecules like NADH and FADH2. Electron transport chain, etc., and oxidative phosphorylation location, inner mitochondrial membrane. Process. The electrons carried by NADH and FADH2 are transferred through the electron transport chain to oxygen, which acts as the final electron acceptor. This process generates a large amount of ATP through chemiosmosis, and water is formed as a byproduct when oxygen combines with electrons and protons. The overall reaction for aerobic respiration is C6H12O6 plus 6O2 right pointing arrow 6 count 2 plus 6H2O plus 36 minus 38 ATP C6H12O6 plus 6O2 right pointing arrow 6 CO2 plus 6H2 O plus 36 minus 38 ATP. Products, CO2, water, and ATP energy. Oxygen required, yes. 2. Anaerobic respiration. Anaerobic respiration occurs in the absence of oxygen. It is less efficient than aerobic respiration and results in the partial breakdown of glucose. Fermentation is the most common form of anaerobic respiration in plants, where pyruvate is converted into ethanol in some plants like yeast or lactic acid in some plant tissues under stress. The 2 main types of anaerobic processes are alcoholic fermentation, common in yeast and some plant tissues. Lactic acid fermentation, less common in plants. Alcoholic fermentation, glucose. Right pointing arrow. 2. Ethanol plus 2 count 2 plus 2 ATP. Glucose right pointing arrow 2 ethanol plus 2 CO2 plus 2 ATP. This occurs mainly in anaerobic conditions when oxygen is absent. Lactic acid fermentation, glucose. Right pointing arrow. 2. Lactic acid plus 2 ATP. Glucose right pointing arrow 2 lactic acid plus 2 ATP. This is a less common process and usually occurs in plant tissues under stress or oxygen-deprived conditions. Products, ethanol or lactic acid, CO2, and a small amount of ATP. Oxygen required, number. While anaerobic respiration provides ATP, it is less efficient than aerobic respiration and is typically used when oxygen is scarce. Photorespiration. Photorespiration is a process that occurs in C3 plants where oxygen is consumed and carbon dioxide is released in the presence of light, leading to a reduction in the overall efficiency of photosynthesis. It is often considered a wasteful process because it reduces the carbon gain and ATP production. Photorespiration occurs when the enzyme Rubisco, which normally fixes CO2 during the Calvin cycle, mistakenly fixes oxygen instead. This happens when CO2 levels are low, typically during hot and dry conditions when stomata close to conserve water, thus reducing the influx of CO2 into the plant. In these conditions, oxygen competes with CO2 for binding with Rubisco. Steps of photorespiration. Initial fixation by Rubisco. Rubisco fixes oxygen O2 instead of CO2, resulting in the formation of phosphoglycolate, a 2-carbon molecule. Formation of glycolate. Phosphoglycolate is then converted into glycolate by a series of reactions. Glycolate to glycine conversion. Glycolate is transported to the peroxisomes where it is converted into glycine, a 2-carbon amino acid. Glycine to serine conversion. In the mitochondria, glycine is converted into serine, releasing CO2 in the process. Return to Calvin cycle. The products are ultimately returned to the Calvin cycle, but this process consumes energy and reduces the plant's overall efficiency in fixing carbon. Photorespiration can be seen as a counterproductive process because it results in the loss of fixed carbon and energy. However, some scientists believe it might also help protect the plant from oxidative damage under certain environmental conditions. Why does photorespiration occur? High oxygen and low CO2. The primary cause of photorespiration is when the CO2 concentration is low and O2 concentration is high, especially under conditions when stomata are closed, e.g., high heat and drought. Rubisco's dual activity. Rubisco has a low specificity for CO2 and O2, meaning it can catalyze both the carboxylation of Rub, which leads to normal photosynthesis, and the oxygenation of Rub, which leads to photorespiration. Consequences of photorespiration. Energy loss. Photorespiration consumes ATP and NADPH without producing any useful sugars or energy for the plant. Carbon loss. Carbon is lost as CO2, which reduces the overall efficiency of photosynthesis. Reduced photosynthetic efficiency. In seed-free plants, photorespiration occurs most often under high light, hot, and dry conditions when CO2 is low. Strategies to overcome photorespiration. Plants in hot and dry climates have evolved different mechanisms to reduce the impact of photorespiration. C4 photosynthesis. In C4 plants, CO2 is initially fixed in mesophyll cells into a 4-carbon compound, which is then transported to bundle sheath cells, where the Calvin cycle occurs, reducing photorespiration. CAM photosynthesis. In CAM plants, CO2 fixation occurs at night when stomates are open, reducing the likelihood of photorespiration during the day. Previous year CSIR net questions. CSIR net June 2020. Question. Photorespiration is a process in plants in which oxygen is consumed and CO2 is released. CO2 is consumed and oxygen is released. Oxygen and CO2 are consumed in equal amounts. Both oxygen and carbon are utilized for the formation of sugars. Answer. Oxygen is consumed and CO2 is released. Photorespiration is a wasteful process where oxygen is consumed and carbon dioxide is released, reducing the efficiency of photosynthesis. CSIR net December 2018. Question. In photorespiration, the initial product formed by the oxygenation of Rub is a phosphoglycerate. B-glyceraldehyde 3-phosphate C-phosphoglycolate phenolate. Answer. C-phosphoglycolate. Photorespiration begins when Rubisco fixes oxygen instead of CO2, leading to the production of phosphoglycolate. CSIR net December 2017. Question. Which of the following is the primary role of Rubisco in plants? A. Oxygen fixation. B. Carbon fixation. C. Protein synthesis. D. Water splitting and photosynthesis. Answer. B. Carbon fixation. Rubisco is primarily responsible for fixing CO2 during the Calvin cycle in photosynthesis. Conclusion. Respiration and photorespiration are critical processes that affect plant energy metabolism and growth. Respiration, whether aerobic or anaerobic, is the process by which plants break down organic molecules to produce energy. In contrast, photorespiration is a less efficient process that can reduce the overall productivity of seed-free plants by wasting carbon and energy. Understanding these processes and underlying biochemical mechanisms is essential for CSIR net JRF life sciences students, especially in the context of plant physiology and metabolic adaptations. You said. Respiration. ChatGPT said. Respiration in plants. Respiration is a crucial metabolic process in plants, responsible for breaking down organic molecules to release energy, which is vital for all cellular activities, growth, and maintenance. Unlike photosynthesis, which stores energy, respiration releases energy stored in glucose and other organic molecules to produce ATP, adenosine triphosphate, the universal energy currency of the cell. Types of respiration in plants. Aerobic respiration. Anaerobic respiration. 1. Aerobic respiration. Aerobic respiration occurs in the presence of oxygen and is the most common and efficient form of respiration in plants. The process involves a complete breakdown of glucose or other organic molecules to produce carbon dioxide, CO2, water, H2O, and a significant amount of ATP. This takes place mainly in the mitochondria of plant cells. Stages of aerobic respiration. Glycolysis. Location. Cytoplasm. Process. Glucose C6H12O6 is partially broken down into two molecules of pyruvate C3H4O3. This process also produces a small amount of ATP, 2-ATP, and NADH, a carrier molecule that will later be used to produce more ATP. Equation. Glucose right pointing arrow. 2. Pyruvate plus 2-ATP plus 2. NADH. Glucose right pointing arrow 2 pyruvate plus 2-ATP plus 2 NADH. Krebs cycle. Citric acid cycle. Location. Mitochondrial matrix. Process. Each pyruvate molecule is converted into acetyl-CoA, which enters the Krebs cycle. In the cycle, acetyl-CoA undergoes a series of reactions, releasing CO2 and transferring high-energy electrons to carrier molecules, NADH and FADH2. A small amount of ATP is also produced. Equation for one acetyl-CoA molecule. Acetyl-CoA plus $3 million plus FAD plus GDP plus pi right pointing arrow county 2 plus 3. NADH plus FADH2 plus ATP. Acetyl-CoA plus 3 NAD plus FAD plus GDP plus pi right pointing arrow CO2 plus 3 NADH plus FADH2 plus ATP. Electron transport chain, etc. and oxidative phosphorylation. Location. Inner mitochondrial membrane. Process. NADH and FADH2, generated in earlier steps, donate electrons to the electron transport chain. This chain of protein complexes transfers electrons, creating a proton gradient across the mitochondrial membrane. The flow of protons back through the enzyme ATP synthase produces a large amount of ATP, about 32 to 34 molecules. Oxygen acts as the final electron acceptor, combining with electrons and protons to form water. Equation. Oxygen plus electrons plus protons right pointing arrow. Water plus ATP. Oxygen plus electrons plus protons right pointing arrow water plus ATP overall equation for aerobic respiration. C. 6H12O6 plus 6O2 right pointing arrow 6 county 2 plus 6H2O plus ATP C 6H12O6 plus 6O2 right pointing arrow 6 CO2 plus 6H2 O plus ATP. Products CO2, H2O, and ATP. Oxygen required. Yes. ATP produced 36 to 38 ATP molecules per glucose molecule. 2. Anaerobic respiration. Anaerobic respiration occurs in the absence of oxygen. In plants, this type of respiration is typically seen under stress conditions, such as during waterlogging or in submerged tissues where oxygen is limited. Anaerobic respiration is less efficient than aerobic respiration, producing much less ATP. Types of anaerobic respiration in plants. Alcoholic fermentation, common in yeasts and some plant tissues. Process. Pyruvate from glycolysis is converted into ethanol and carbon dioxide, with the regeneration of NAD+ required for glycolysis to continue. This process produces a small amount of ATP, 2-ATP per glucose molecule. Equation. Glucose right pointing arrow. 2. Ethanol plus 2 county 2 plus 2 ATP. Glucose right pointing arrow 2 ethanol plus 2 CO2 plus 2 ATP. Lactic acid fermentation, less common in plants but can occur under extreme anaerobic conditions. Process. Pyruvate is converted into lactic acid. Like alcoholic fermentation, it regenerates NAD+ but does not produce any ATP beyond the initial glycolysis. Equation. Glucose right pointing arrow. 2. Lactic acid plus 2 ATP. Glucose right pointing arrow 2 lactic acid plus 2 ATP anaerobic respiration summary. Products. Ethanol in alcoholic fermentation or lactic acid in lactic acid fermentation, CO2 and 2 ATP per glucose. Oxygen required. Number. ATP produced. 2 ATP per glucose molecule, far less efficient than aerobic respiration. Key differences between aerobic and anaerobic respiration. Feature aerobic respiration. Anaerobic respiration oxygen requirement requires oxygen, does not require oxygen. ATP produced for glucose 3638 ATP 2 ATP byproduct CO2 and H2O ethanol and lactic acid. CO2 efficiency high, complete breakdown of glucose, low partial breakdown of glucose, regulation of respiration. Respiration in plants is regulated by several factors. Oxygen availability. In aerobic conditions, oxygen acts as the final electron acceptor in the electron transport chain, allowing the full breakdown of glucose. In low oxygen conditions, anaerobic pathways are activated. Temperature. Temperature affects the rate of enzyme catalyzed reactions in respiration. Higher temperatures increase the rate of respiration up to an optimal point, beyond which enzymes may denature, slowing the process. Hormonal control. Hormones like ethylene play a role in regulating respiration during processes such as fruit ripening or seed germination, where respiration rates increase. Nutrient availability. The availability of glucose and other substrates affects the rate of respiration. Nutrient limitation, e.g., glucose, nitrogen, can limit respiration. Previous year CSIR net questions. CSIR net June 2020 question. During aerobic respiration, the oxygen consumed is used in a glycolysis. B. Krebs cycle. C. Electron transport chain defermentation. Answer. C. Electron transport chain. Oxygen acts as the final electron acceptor in the electron transport chain during aerobic respiration. CSIR net December 2018 question. In anaerobic respiration in yeast, the end product is a lactic acid. B. Ethanol. C. Oxygen deacetate. Answer. B. Ethanol. In the absence of oxygen, yeast undergo alcoholic fermentation, producing ethanol and CO2. CSIR net June 2017 question. The majority of ATP produced during aerobic respiration is generated by a glycolysis. B. Citric acid cycle. C. Oxidative phosphorylation defermentation. Answer. C. Oxidative phosphorylation. The majority of ATP is produced during oxidative phosphorylation in the electron transport chain. Conclusion. Respiration is an essential metabolic pathway that enables plants to convert stored energy in organic molecules into usable energy, ATP. Aerobic respiration is the most efficient process, generating a significant amount of ATP through glycolysis, the Krebs cycle, and oxidative phosphorylation. In contrast, anaerobic respiration is less efficient but important when oxygen availability is limited. Understanding the processes of respiration and its regulation is critical for CSIR net JRF life sciences preparation, especially in the context of plant physiology. You said, no table ChatGPT said, respiration in plants. Respiration is a crucial metabolic process in plants, responsible for breaking down organic molecules to release energy, which is vital for all cellular activities, growth, and maintenance. Unlike photosynthesis, which stores energy, respiration releases energy stored in glucose and other organic molecules to produce ATP, adenosine triphosphate, the universal energy currency of the cell. Types of respiration in plants. Aerobic respiration. Anaerobic respiration. 1. Aerobic respiration. Aerobic respiration occurs in the presence of oxygen and is the most common and efficient form of respiration in plants. The process involves the complete breakdown of glucose or other organic molecules to produce carbon dioxide, CO2, water, H2O, and a significant amount of ATP. This takes place mainly in the mitochondria of plant cells. Stages of aerobic respiration. Glycolysis. Location. Cytoplasm. Process. Glucose C6H12O6 is partially broken down into two molecules of pyruvate C3H4O3. This process also produces a small amount of ATP, 2 ATP, and NADH, a carrier molecule that will later be used to produce more ATP. Equation. Glucose right pointing arrow 2 pyruvate plus 2 ATP plus 2 NADH. Krebs cycle. Citric acid cycle. Location. Mitochondrial matrix. Process. Each pyruvate molecule is converted into acetyl-CoA, which enters the Krebs cycle. In the cycle, acetyl-CoA undergoes a series of reactions, releasing CO2 and transferring high-energy electrons to carrier molecules, NADH and FADH2. A small amount of ATP is also produced. Equation for one acetyl-CoA molecule. Acetyl-CoA plus $3 million plus fat plus GDP plus pi right pointing arrow CO2 plus 3 NADH plus FADH2 plus ATP. Electron transport chain, etc., and oxidative phosphorylation. Location. Inner mitochondrial membrane. Process. NADH and FADH2, generated in earlier steps, donate electrons to the electron transport chain. This chain of protein complexes transfers electrons, creating a proton gradient across the mitochondrial membrane. The flow of protons back through the enzyme ATP synthase produces a large amount of ATP, about 32 to 34 molecules. Oxygen acts as the final electron acceptor, combining with electrons and protons to form water. Equation. Oxygen plus electrons plus protons right pointing arrow water plus ATP. Overall equation for aerobic respiration. C6H12O6 plus 6O2 right pointing arrow 6CO2 plus 6H2O plus ATP. Products. CO2, H2O, and ATP. Oxygen required. Yes. ATP produced. 36 to 38 ATP molecules per glucose molecule. 2. Anaerobic respiration. Anaerobic respiration occurs in the absence of oxygen. In plants, this type of respiration is typically seen under stress conditions, such as during waterlogging or in submerged tissues where oxygen is limited. Anaerobic respiration is less efficient than aerobic respiration, producing much less ATP. Types of anaerobic respiration in plants. Alcoholic fermentation, common in yeast and some plant tissues. Process. Pyruvate from glycolysis is converted into ethanol and carbon dioxide, with the regeneration of NAD+ required for glycolysis to continue. This process produces a small amount of ATP, 2 ATP per glucose molecule. Equation. Glucose right pointing arrow to ethanol plus 2 CO2 plus 2 ATP. Lactic acid fermentation, less common in plants but can occur under extreme anaerobic conditions. Process. Pyruvate is converted into lactic acid. Like alcoholic fermentation, it regenerates NAD+ but does not produce any ATP beyond the initial glycolysis. Equation. Glucose right pointing arrow to lactic acid plus 2 ATP. Anaerobic respiration summary. Products. Ethanol in alcoholic fermentation or lactic acid in lactic acid fermentation, CO2, and 2 ATP per glucose. Oxygen required. Number. ATP produced. 2 ATP per glucose molecule, far less efficient than aerobic respiration. Key differences between aerobic and anaerobic respiration. The main differences between aerobic and anaerobic respiration in plants are related to oxygen requirement, ATP production, and the byproducts generated. In aerobic respiration, oxygen is required and results in a much higher ATP yield, 36 to 38 ATP per glucose molecule. In contrast, anaerobic respiration occurs without oxygen and generates only 2 ATP per glucose molecule, with byproducts like ethanol or lactic acid. Regulation of respiration. Respiration in plants is regulated by several factors. Oxygen availability. In aerobic conditions, oxygen acts as the final electron acceptor in the electron transport chain, allowing the full breakdown of glucose. In low oxygen conditions, anaerobic pathways are activated. Temperature. Temperature affects the rate of enzyme catalyzed reactions in respiration. Higher temperatures increase the rate of respiration up to an optimal point, beyond which enzymes may denature, slowing the process. Hormonal control. Hormones like ethylene play a role in regulating respiration during processes such as fruit ripening or seed germination, where respiration rates increase. Nutrient availability. The availability of glucose and other substrates affects the rate of respiration. Nutrient limitation, e.g., glucose, nitrogen, can limit respiration. Previous year CSIR net questions. CSIR net June 2020 question. During aerobic respiration, the oxygen consumed is used in a glycolysis. B. Krebs cycle. C. Electron transport chain defermentation. Answer. C. Electron transport chain. Oxygen acts as the final electron acceptor in the electron transport chain during aerobic respiration. CSIR net December 2018 question. In anaerobic respiration in yeast, the end product is a lactic acid. B. Ethanol. C. Oxygen deacetate. Answer. B. Ethanol. In the absence of oxygen, yeast undergo alcoholic fermentation, producing ethanol and CO2. CSIR net June 2017 question. The majority of ATP produced during aerobic respiration is generated by a glycolysis. B. Citric acid cycle. C. Oxidative phosphorylation defermentation. Answer. C. Oxidative phosphorylation. The majority of ATP is produced during oxidative phosphorylation in the electron transport chain. Conclusion. Respiration is an essential metabolic pathway that enables plants to convert stored energy in organic molecules into usable energy, ATP. Aerobic respiration is the most efficient process, generating a significant amount of ATP through glycolysis, the Krebs cycle, and oxidative phosphorylation. Aerobic respiration is the most efficient process, generating a significant amount of ATP through glycolysis, the Krebs cycle, and oxidative phosphorylation. In contrast, anaerobic respiration is less efficient but important when oxygen availability is limited. Understanding the processes of respiration and its regulation is critical for CSIR net JRF life sciences preparation, especially in the context of plant physiology. You said, photorespiration. ChatGPT said, photorespiration in plants. Photorespiration is a metabolic process that occurs in plants, where the enzyme riboscope ribulose bisphosphate carboxylase oxygenates rub ribulose bisphosphate instead of carboxylating it, leading to the formation of 2-phosphoglycerate rather than 2-phosphoglycerate. This process is considered wasteful because it results in the loss of fixed carbon and energy, and it reduces the efficiency of photosynthesis. Photorespiration is a response to high oxygen O2 levels and low carbon dioxide CO2 concentrations. Mechanism of photorespiration. Photorespiration occurs under conditions where the oxygen concentration is higher than the carbon dioxide concentration, typically in hot and dry environments where plants close their stomata to conserve water. This causes a buildup of O2 inside the leaf and a decrease in CO2, which increases the likelihood of rubisco binding with O2 instead of CO2. Steps of photorespiration. Oxygenation of rub by rubisco. Rubisco acts as both a carboxylase fixing CO2 and an oxygenase fixing O2. When oxygen is abundant, rubisco catalyzes the reaction between rub and O2, producing 3-phosphoglycerate 3-PGA and 2-phosphoglycolate. The 3-PGA is a normal product of photosynthesis, but 2-phosphoglycolate is a waste product that cannot enter the Calvin cycle. Equation. Rub plus O2 right pointing arrow 3-PGA plus 2-phosphoglycolate. Conversion of 2-phosphoglycolate. The 2-phosphoglycolate formed is toxic to the plant and must be processed. It is transported from the chloroplast to the peroxisome, where it is converted into glycolate by a series of reactions. Glycolate is then converted into glycine in the mitochondria, and 2 molecules of glycine combine to form serine, releasing CO2 in the process. Release of CO2. The final step of photorespiration involves the release of CO2 as a result of the degradation of glycine to serine. This CO2 is lost to the atmosphere and does not contribute to the plant's photosynthetic carbon fixation. Overall equation of photorespiration: 2. Glycine right pointing arrow. Serine plus CO2. 2 glycine right pointing arrow serine plus CO2. This process ultimately reduces the efficiency of photosynthesis as it leads to the release of fixed carbon. Significance of photorespiration: energy loss. Photorespiration consumes ATP and NADPH without contributing to the production of sugars. This results in an overall loss of energy in the form of ATP and a reduction in photosynthetic efficiency. Carbon loss. The main issue with photorespiration is that it leads to the loss of fixed carbon CO2, which could have been used in the Calvin cycle to produce glucose and other carbohydrates. Occurs in high O2 and low CO2. Photorespiration is more prominent in conditions where there is high oxygen O2 concentration and low carbon dioxide CO2 concentration, typically when the plant's stomata are closed to prevent water loss during hot and dry conditions. Photorespiratory cycle. The photorespiratory cycle also involves several organelles, such as chloroplasts, peroxisomes, and mitochondria, to process the byproducts. This complexity is one reason why photorespiration is less efficient than regular photosynthesis. Photorespiration versus photosynthesis. While photosynthesis involves the fixation of carbon dioxide into sugars, photorespiration is a side reaction that competes with the Calvin cycle, leading to carbon loss. Both processes involve rubisco, but while photosynthesis is beneficial and produces sugars, photorespiration consumes energy and results in a net loss of carbon. Photosynthesis. CO2 plus H2O right pointing arrow glucose plus O2 energy storing process. Photorespiration. Rub plus O2 right pointing arrow 3-PGA plus 2-phosphoglycolate wasteful process, releasing CO2. Factors influencing photorespiration: oxygen concentration. Higher oxygen concentrations increase the likelihood of rubisco acting as an oxygenase rather than a carboxylase, leading to photorespiration. This is more common in hot conditions when plants close their stomata to reduce water loss, limiting CO2 uptake. Carbon dioxide concentration. Lower CO2 concentrations favor photorespiration since rubisco has a higher affinity for oxygen under such conditions. This is particularly relevant when stomata are closed and CO2 cannot enter the plant tissues. Temperature. High temperatures can increase the rate of photorespiration because of the increased oxygenation reaction by rubisco. This is one of the reasons why plants growing in hot climates may suffer from decreased photosynthetic efficiency during the day. Adaptations to reduce photorespiration. Plants have evolved several mechanisms to minimize photorespiration, especially in hot and dry environments. C4 photosynthesis. C4 plants, such as maize and sugarcane, use a different pathway to concentrate CO2 around rubisco, effectively reducing the chances of oxygen interacting with rub. This reduces photorespiration and increases the efficiency of photosynthesis. CAM photosynthesis. Crassulation acid metabolism. CAM plants, like cacti and succulents, open their stomata at night to fix CO2 into organic acids. This allows them to reduce photorespiration by maintaining high CO2 concentrations during the day when stomata are closed to conserve water. Rubisco modification. Genetic engineering efforts have aimed at improving rubisco specificity for CO2 over O2 to reduce photorespiration. Some plants have evolved or are being developed with modified versions of rubisco to enhance photosynthetic efficiency. Previous year CSIR net questions. CSIR net June 2020 question. Photorespiration in plants leads to the release of oxygen. B nitrogen, C carbon dioxide, D methane. Answer: C carbon dioxide. Photorespiration releases carbon dioxide as a waste product, reducing the efficiency of photosynthesis. CSIR net December 2019 question. In photorespiration, the product that is directly processed in the peroxisome is A 3-phosphoglycerate. B glycolate, C glycine, D serine. Answer: B glycolate. Glycolate is transported to the peroxisome, where it is converted into glycine. CSIR net June 2018 question. The enzyme involved in photorespiration is a rubisco. B-PAP carboxylase C-ATP plus reductase C-ATP synthase. Answer: a rubisco. Rubisco catalyzes both the carboxylation and oxygenation reactions during photosynthesis and photorespiration. Conclusion. Photorespiration is a metabolic process that reduces the efficiency of photosynthesis by consuming energy and releasing CO2, thus limiting plant growth and productivity, particularly in conditions of high oxygen and low CO2.